So I think most people in the room are probably familiar with metagenomic sequencing or next generation sequencing, but I'm just going to give a quick overview. So why do we want to use next-gen sequencing? We've been using Sanger sequencing for a long time. Really answers data. Sanger sequencing, which we all do, which we all love, generates a read about 800 bases. When we send it out to third parties, it's about $5 to sequence, so it's cheap. Next-gen sequencing, however, for a sample, typically when we run samples at KSVDL, we generate about 1 million reads on a sample. The reads are a little bit shorter, about 300 bases, but you can obviously do the math. There's a whole lot more there. The other real important part of this is when we do Sanger sequencing, we need to know what we're looking for. So we need to design a primer up front, and then sequence, again, what we're expecting. With next-generation sequencing, we don't need to know. So this is really the real main benefit of next-gen sequencing. And it's really unique to this technology. There's a lot of different diagnostic assays that have been developed. We can use pan family primers. But again, you always need to have some prior information on what you're looking for. Here, absolutely, we don't need to know. And then in terms of economics, it's great. These technologies are really cool, but if we can't afford to run them, there's no point in terms of practical day-to-day use. So at KSVDL, Sanger sequencing for PRRS 5 is $182 for 600 bases, so 30 cents a base. Next-gen sequencing, we can do a full PRRS genome for $300, so 2 cents a base. So again, what we do is we generate a whole lot more information that we can mine for important information in terms of epidemiology and vaccine design. The other beauty of this is, again, using the PRRS example, if we're trying to sequence PRRS, we can also see what else is in that sample. So not just quasi-species of PRRS, but other viruses that are infecting the animal. So again, those are kind of the reasons why we want to do next-gen sequencing. In particular, we use a method called metagenomic sequencing. So here, we're sequencing material directly from the field. So a lot of times with pigs, it's serum samples, nasal swabs, tissues, whatever. Previously we would isolate virus on cells, grow them up, then do PCR, and then sequence. Here, sample comes in from the field, and we're sequencing it directly. So this is a quick overview of how we do this. We start with the sample, and we pre-treat it with a cocktail of DNases and RNases. And the idea here is the nucleic acids for the viruses are protected by the viral capsid. So we're degrading host and an unprotected DNA and RNA. We then randomly reverse transcribe nucleic acids with a primer. Essentially, it's got a random hexamer. So random priming with a 20 base pair barcode attached to it. We do reverse transcription, second strand synthesis, and then we amplify that because, again, a lot of these viruses are present at very low levels because, again, we're sequencing from the environment. So we use that barcode sequence specifically to amplify the DNA. And then at the end of the day, what you have is a pool of PCR products representing everything that's in that sample. So I'm going to talk more about Parainfluenza virus in a little bit. But this is just an example of how we do the data analysis. So this was a sample that came in the lab. We did metagenomic sequencing, analyzed all the fragments that were present. We saw hits for Parainfluenza 1 virus. So we went back and downloaded a reference genome, which is illustrated up on the top of that figure. And then we mapped the reads to that sample. So all the little the rainbow of lines below, in the middle and the bottom part of the slide are individual reads. So for this particular sample, we had 11 million reads. Only 52,000 mapped to parainfluenza virus, so 0.45%. So you might think, well, that's really kind of not that good. It's a very small percentage of the reads. However, because we have so much depth, we had 361x coverage of the genome. So again, from a nasal swab from a pig with acute respiratory disease, we were able to pull out a full parainfluenza virus genome. So I'm going to start here with the first example I'm going to talk about is using next-gen sequencing for PRRS epidemiology. So this all started with the pork-board grant we had last year, which the objective was, can we sequence PRRS directly from samples from the field. We originally tried oral fluids and nasal swabs, and we were able to get PRRS sequence, but not complete genomes. Really, I think it's a reflection of the amount of virus in oral fluids and nasal swabs. We just don't have the titer that you do in serum. So we ended up switching over to serum. We got pretty good results. I'm not sure how this works. Can we go back a slide? Sorry. So figure 1 there is looking at PRRS-specific reads as a function of CT value. So as you can imagine, the more virus we have, the larger the percentage of the reads that are going to map back to PRRS. So basically, we had a CT value of 15, so obviously very strong viremia. Essentially all the reads that we were sequencing in serum were PRRS. And on figure 2, you could see when we had a CT value of 20 or lower, we were able to get complete PRRS genome coverage. But even out to a CT of 25, we're getting the vast majority of the genome. So again, with samples coming to the lab, finding a CT value of 25 or less is not that difficult. So the method looked pretty good when we were doing development work. So we next applied it to 182 total PRRS positive serum samples. We were able to pull out 66 complete genomes. A lot of the samples where we did not get genomes had higher CT values, and again, we just took random samples. And the other thing we deal with is co-infections. With respect to PRRS specifically, a lot of the samples where we were not able to get PRRS genomes, we had a lot of parvoviruses, parvo2 in particular, and parvo6. So again, if you have a population of viruses, and if there's a much higher titer of parvo, that really ends up eating up a lot of the reads. I'm not going to get into the PRRS phylogeny into much depth here. But again, so we were able to determine 66 complete genomes. It's the phylogenetic analysis of GP2 through GP5. For each of those genes, we're able to find 4 to 7 well-supported clades, less than 10% diversity within a clade, greater 10% interclade diversity. On the top right-hand side, you can see I marked RFLP 174. I know later we're going to hear some talks about 174. So we did find some 174s there. But the one thing I wanted to bring up here was topological incongruences. So the standard way to do PRRS epidemiology is to sequence GP5. Over the years, literally tens of thousands of GP5 sequences have been derived from various labs. And we use that as a basis to determine which strains are going where. You can see in pink there, we have a number of viruses when we assign just basically allele identifiers for the various clades of GP5. You can see there's a number of viruses that had more or less very similar GP5 genes, but if you look at GP2, GP3, and GP4, you can see different allelic combinations. So it really begs the question, is GP5 the right target to be looking at? PRRS is known to recombine readily, and looking at this, again, you're going to derive a different conclusion depending upon which gene you're looking at when you do phylogeny. So what I would argue is it's a much better approach to do full genome sequencing, especially now that it's becoming much simpler and more affordable. The other thing we looked at in these viruses was NSP2. NSP2 is important for viral pathogenicity and is highly variable. One of the things we noticed right away was a number of unique NSP2 deletions. So on the tree here, at the bottom are viruses that were, NSP2s are very much like VR2332. Above that we had a clade, a well-supported clade, with a large single novel deletion that we could not find any evidence for previous description. What was interesting, these were all RFLP 174 strains. And again, RFLP 174 is determined based on GP5. Above that in blue, the big box, are all strains with Minnesota 184-like NSP2 deletions. So one of the features of Minnesota 184, besides its increased pathogenicity when it was first identified, was it had 3 discontinuous deletions in NSP2. So we had a clade of viruses there that are RFLP 174 that had NSP2s very much like Minnesota 184. But then above that in the box, we had another RFLP 174 strain that had 4 deletions in NSP2. So When we went back and looked specifically at the RFLP 174 strains, at the structural genes, the GP2A, 3, 4, 5, M, and N, we saw that the RFLP 174 viruses all had essentially the same alleles, and the variability was very small, greater than 95% identity for those genes. However, if we look at NSP2, we see a tremendous amount of diversity in RFLP 174 strains. So again, what does it mean? I honestly don't have an answer for you, but it could be involved with differing clinical presentations for RFLP 174, because again, our NSP2 has been shown to affect viral pathogenicity. So this slide attempts to summarize some of the reasons why I think we should consider moving from GP5 to full genome sequencing. Again, GP5 is the major surface glycoprotein. That's why it was chosen as a target early on. It's highly variable, has neutralizing epitopes. However, a lot of work's been done looking at the immune response to GP5, and it's been shown to the antibodies directed towards GP5 arise mostly after the clearance of viremia. Previous work has shown that glycoprotein 2A, 3, and 4 are important for receptor binding proteins, as well as have neutralizing epitopes and B-cell epitopes. So we heard earlier, again, the effect of the CD163 deletion in pigs. So again, very important components of the virion that we're largely ignoring by just sequencing GP5. And also on the last slide, I mentioned phylogenetic incongruence. If you're doing epidemiology, what do you believe? There's a lot of papers looking at literally thousands of GP5 sequences, but if I was to do the exact same work with GP2A or GP3, essentially I'd derive a different conclusion. Again, NSP2 is important for pathogenicity, and again with the RFLP 174 strains, we see quite a bit of genetic variability as well as unique deletions. And the other thing is, again, as I mentioned earlier, we can detect other viruses when we do this approach. So on the bottom there are two papers we published, which were viruses we found as part of this porkboard project. So again, we were sequencing serum samples trying to derive full PRRS genomes. The bottom right, we had the first identification of porcine parvovirus 6 in North America. So this was a parvovirus that was described earlier this year in China. And this was actually fairly common in our data set. And then on the bottom left, discovery of a novel Pestivirus that I'm going to talk about next. So when we were sequencing these samples, again, going through the contigs, as we generate these 1 million reads per sample, for example, we assemble these in the contigs and we blast them out looking to identify what's in the sample. We had one sample, we had a number of hits coming back: classical swine fever, BVD, border disease. However, the expectation values were pretty low. So it was clear we did not have classical swine fever. But we had a novel Pestivirus. So we were able to assemble a complete genome, and essentially identified a very highly divergent Pestivirus. On an overall level, it's very distantly related to BVD and classical swine fever, so about 25% identity. What it was most closely related to was a bat Pestivirus that was partially described from China, partially meaning that the paper they identified in China had about 5 kb of the genome, and it's about a 12 kb genome. So, however, over that 5 kb region, we had about 68% identity. In our sample set, we had 5 out of 182 samples where we could identify this Pestivirus by sequencing, which was subsequently confirmed by real-time PCR. So this is this chart here shows the various proteins of the Pestivirus and their best blast hits. So RAPV is Rhinolaphis affinis Pestivirus. So this is the bat Pestivirus. So again, where we had bat Pestivirus sequence, we had the highest percent identity. So again, overall for the bat, it was about 68% identity. But what was interesting, proteins such as NPRO, which is an autoprotease which is unique to Pestiviruses, by BLAST-P has no homology to anything in GenBank. So obviously a very divergent virus. E2 is very important for receptor binding for pestiviruses. What's interesting about E2 for this virus as well as the bat pestivirus is they're considerably shorter than other pestiviruses, on the order of over 100 amino acids are deleted from that protein. So again, very unique virus. Phylogenetically, again, where we have that pestivirus sequence, you can see a close association with our pestivirus. So up in A for the complete polyprotein, and in D there we're looking at NS3. So again, close relationship. And again, these viruses are very distantly related to conventional pestiviruses such as BVD, classical swine fever. I mentioned we had 5 samples that were positive for this pestivirus, which is confirmed by PCR. We had 2 different PCRs. The initial one was targeting E2. The second one was targeting the ERNS region of the genome. 3 of the 5 were positive on the ERNS assay. 1 sample we didn't have enough of it, so we couldn't test it for the ERNS. And then we had 1 sample that was only positive on the ERNS, but was negative on the E2. We went back to that sample and sequenced it. And we found that the E2 was only 88% identical at the amino acid level. So again, what this suggests is this is not a single strain, that's highly homologous. Essentially, we have a fair amount of genetic diversity in this pestivirus. And in terms of distribution of where these 5 positives came from, Nebraska, Arizona, North Carolina, Minnesota, and Kansas. So certainly suggesting this thing is widely distributed in the U.S. Dr. Yingfang's group generated recombinant ERNS protein in E. coli, and we used that to run ELISA on serum samples. Originally, when we did this, I didn't mention this earlier, but we originally found this in PRRS positive samples. We went back and also screened 300 PRRS negative samples, and we could not find the virus in PRRS negative samples. So we were thinking that this virus was associated with PRRS. So when we did the serology, we tried to differentiate the status of the sample with respect to PRRS. So we ran 78 samples that were PRRS PCR positive, 94% were positive. And then we also looked at 90 samples from a single farm that was PRRS antibody positive. 100% of the samples were positive. We went to a PRRS antibody negative farm, ran 48 samples, and again had a very high positive rate, 96%. However, PRRS antibody farm 2 was totally negative for the pestivirus. So clearly not a perfect association or correlation with PRRS in the presence of this pestivirus. And as far as controls, age-matched specific pathogen-free pigs were negative, as well as the second PRRS antibody negative farm. That was the initial description of the virus. We published that in General Virology this summer. But then back in June, I had a call from a veterinarian out in North Carolina who had approximately 700 pigs that had died with what he called intention tremors. So pigs were about 5 weeks of age when this began. Pigs would start to shake, have tremors. It would progress to the point where the animal couldn't stand, couldn't swallow, 100% mortality. So as pigs would die, new pigs in that group would develop these symptoms and die. So this went on out to about 14 weeks of age. And again, 100% mortality. So those samples were sent to us. We did metagenomic sequencing on the brain. And the only virus we could identify was our new pestivirus. And again, we only had 195 reads. But those reads had about 87% identity to the pestivirus we had previously published. We did follow-up PCR on brain, lymph nodes, heart, and serum. All these were positive. The lowest CT values were in the lymph nodes. And then in August, essentially I got a call from the vet again. Now this was a successive batch of pigs that had originated from the same sow farm. These pigs were now at 10 to 16 weeks of age. Started seeing the same clinical presentation, sent samples in. We went straight to the PCR this time and confirmed the pestivirus. So this is a figure from the second outbreak, or a movie from the second outbreak. The clinical symptoms, what they were seeing. All right. So obviously it's fairly significant clinically. Dr. Yingfeng's group also generated polyclonal as well as monoclonal antibodies against the ERNS protein in mice. So the top figure are basically the controls. So she had MARC145 cells expressing ERNS. You can see nice fluorescence. And down in C, you can see a lymph node from one of the APPV-infected pigs showing positive staining for this virus. Down below that, we also looked at other tissues, IHC positives in the liver, lymph node and spleen. Again, we had better staining in the lymph node, and that corresponds to what we see with PCR. It seems like there's lymph nodes are the preferred site of replication for this virus. We were unable to find the virus in the brain, but then going back to the PCR, we have very low levels in the brain. CT value is about 35. We have a hard time pulling it out with sequencing in the brain. We just get partial sequences. So really, in terms of where you look for this thing, lymph nodes, at least now, I would say, are probably the best bet. Iowa State also was on the trail of this Pestivirus. They issued a press release a while back here. And basically said that they had identified a Pestivirus in pigs born with congenital shaking. And they went back, took that sample, inoculated fetuses in utero, and were able to reproduce disease. Talking to ISU, the Pestivirus that they have on a nucleotide level is 88% similar to the one we have. So I think, again, consistent with it being the same species of virus, but again, as similar to what we saw, there is a fair amount of diversity in this Pestivirus. So the big question is really, how serious is this disease to the swine industry? I don't have a good answer. Congenital tremors have been around for a long time. People have seen these sporadically. And there's been lots of papers and investigations and putative virus X's that people have seen, but really no definitive etiological agent until now. So we know about congenital tremors, but the intention tremors in the older pigs, I have personally not heard that much about. Again, this was a fairly big outbreak. 700 heavy hogs died. So I don't know if this is a new clinical presentation from an old virus, or if the virus has changed. But then now, again, this is a very novel Pestivirus. We have a lot of questions. You know, Pestiviruses tend to cause persistent infection, so can we get persistent infection in pigs? I mentioned the diversity earlier. Do we have different clinical presentations with different variants of the virus? And then again, the other question, is this something that's new to the U.S. or something that's been here for a long time? Serologically, it's fairly widespread. And again, if this is the sole agent involved with congenital tremors, I think that would certainly suggest this has been here for a long time and it's been unnoticed. So now I'm going to have a few slides here on this Parainfluenza virus 1. Parainfluenza virus 1 was described in Hong Kong in 2013. The group out there was doing metagenomic sequencing on pigs that arrived deceased at a slaughterhouse. So in the same week, I had two different veterinarians call me about young pigs, so 10 to 21 days of age with acute respiratory disease. PCRs for flu and PRRS as well as bacteria were negative. So they were curious if we could sequence to see what was going on in those animals. We did metagenomic sequencing directly on the nasal swabs or nasal turbinates. And we're able to assemble two complete genomes, one from Illinois, one from Nebraska. You can see them there. So the two U.S. viruses are very similar to the three described in Hong Kong. And again, that clade is well defined, most similar to human parainfluenza virus 1 and Sendai virus. At an overall genetic level, about 90 to 95% identity in the U.S. to the Hong Kong viruses. We also designed a real-time PCR and screened additional samples and took a bunch of those and just did HN gene sequencing. And you can see there, again, we have a well-defined clade, positive samples from Nebraska, Illinois, and Oklahoma. The HN genes specifically had a 85 to 96% identity to those from Hong Kong. So again, suggesting there's a fair amount of diversity in these parainfluenza viruses in the United States. We used that real-time PCR to look at the incidence of parainfluenza virus 1. 17 out of 279 samples were positive. And these were lung homogenates, oral fluids, and nasal swabs submitted for respiratory disease testing. So more than likely they were submitted for flu or PRRS. So 6.1% were positive. We set up 2 different serological assays. One, the recombinant F protein ELISA. Again, Dr. Fong's lab generated recombinant protein and made antibodies for us. And we had 63% of the serum samples positive. We also developed an immunocapture PCR-based serological assay. And had 55% positive that assay. So again, fairly consistent results. We did in situ hybridization on approximately 3-week-old pig that was naturally infected with parainfluenza virus 1. We saw a lot of staining in the nasal turbinates and just real sporadic staining in the trachea. We were unable to identify the virus in the lung. So again, it would appear that the site of replication is the upper respiratory tract, specifically the respiratory epithelial cells in the turbinates. So to summarize the parainfluenza virus, I would say this virus is fairly common in U.S. pigs based on the limited diagnostic testing we've done. Talking to veterinarians in the field, this is causing significant respiratory disease, typically in younger pigs. Pigs 10 to 14 days of age, I would say, are probably the most common. And again, it's something that veterinarians need to be aware of and to include in differential diagnostics for flu and PRRS. Again, K-State, we'll plug K-State here, has a PCR setup for parainfluenza virus. And I've had a number of people calling, asking about vaccines, asking, are vaccines available? Can you isolate this virus? Can we do autogenous? Because if it was available, we would vaccinate. So again, I take that as this is fairly significant from the field if people are asking and wanting to vaccinate pigs. Unfortunately, nothing is available at this point. The next topic, Seneca Valley virus. So we had a project last summer that was funded by Zoetis as well as BI. And what we were doing is metagenomic sequencing of fecal nasal swabs from swine markets in North Carolina. And this was a summer project for a North Carolina State second-year vet student. So this was her internship project. She collected the sample, sent them to us for sequencing. So this was purely a bug hunt. This was swabs were pulled from slaughterhouses, from primary markets, so high health pigs at slaughter. We also pulled samples from cull, secondary market slaughterhouse pigs, and then we pulled samples from a cull pig buying station. In terms of the samples, we had 5 pigs per producer sampled per site, and again, 1 nasal, 1 fecal swab. So 5 producers per site. We pooled swabs. So we ended up running 50 pools. And we did this twice. The first one was, the first sampling was supported by Zoetis. The second sampling was supported by BI. In terms of what we found, I'm going to leave this for Olivia since it was her project. She's presenting this as an oral presentation at AASV this coming spring. But it really was virus soup. I know in the second sample set we identified over 30 viruses. And if you can think of a swine virus in the U.S., we found it. But what was really interesting and unexpected was that we identified Seneca Valley virus in both samplings from 4 out of 5 of these markets. And again, this was identified by deep sequencing. So we went back and did real-time PCR to confirm the sequencing results. 26 out of 50 pools were positive in our original sampling. In the second sampling, 18 out of 50 samples. But what was really interesting is when we broke those results into primary versus secondary market pigs. So again, the high health pigs at the primary markets, we had 1 positive, 2.5%. Secondary market, 72% of the pigs were positive. We did attempt virus isolation on 5 of the samples that had lowest CT values, and they were all positive. The virus grew well, CT values 15 to 20. And what was interesting about this data set was really the contrast to information we heard from the Swine Health Information Center. They had supported a large number of oral fluids to be tested at ISU in Minnesota for Seneca Valley and reported 1% positive, which again is in line with our primary market samples. However, again, looking at the secondary market, we saw much higher prevalence. So, it really begs the question, what does this mean? Is Seneca Valley more common in these lower health status pigs just because they're immunocompromised? Or does Seneca Valley play some role in those pigs being of a lower health status? It's kind of a chicken versus egg argument, but it's pretty interesting. We were able to assemble 5 complete genomes directly from the swabs. You can see them in the top right, North Carolina 2015. The virus was most similar to ones that Minnesota published from Brazil in 2015. And both of these viruses have a common ancestor in this Canadian 2011 Seneca Valley virus. All the U.S. genomes, greater than 99% identity, and again, very similar to the Brazilian as well as Canadian Seneca Valley, and more dissimilar from historical U.S. viruses. The final topic is a new circovirus, and this is a relatively recent result. We haven't published this yet. And again, it started with a veterinarian calling me from North Carolina, basically. Sow farm, chronic poor reproductive performance. Sow is acutely dying with PDNS-like symptoms. Samples originally went to ISU. Histopathologically, everything looked like PCV2. However, PCR and IHC were negative for PCV2. Samples were forwarded to us for metagenomic sequencing. The arrow shows the complete genome of the virus on the phylogenetic analysis. We're calling this PCV3. You can see down at the bottom is PCV1 and 2. So this virus is very distantly related to PCV1 and 2. On the left is a cartoon of the genomic architecture. So again, a capsid and replicase. It's about 200 bases than PCV2. But it's interesting, the replicase shares only 54% identity to a bat circovirus. The capsid shares only 36% identity to PCV2. So again, this is a fairly divergent novel circovirus. We found an additional ORF that had 39% identity to a mirrored herpesvirus protein of unknown function. So again, fairly unique virus. We went on to screen samples looking for incidence of this virus in the field. 12.5% were positive. We also went back to the original samples to quantify how much virus is there. Sows with PDNS that had died acutely with PDNS-like lesions had a CT 28 to 30. The mummies that were submitted from sows that died with PDNS-like lesions had a CT 17 to 21. So there was a lot of this virus in the aborted tissues. And again, so we find a lot of viruses, and the question and a lot of times the criticism of metagenomic sequencing is, how do we know what's important? This one I would say is unique and probably being important. If you think about the history of PCV2, again, PCV2 was first identified in the 90s. In Canada, sporadic PMWS, progressed to widespread epidemics, Europe, Asia, followed by the United States, and really was uncontrolled until we had commercial vaccines in 2005. So really the question here is, we've got a very novel circovirus causing apparently sporadic clinical disease. Is this virus on a similar trajectory to PCV2? So obviously that's pure speculation. We need to do more research. So last slide, just to summarize next-gen sequencing, metagenomic sequencing. It really is a powerful tool, and I hope these examples illustrate the power of this. These are viruses we found in the last year just by running samples. It's good for viral genome sequencing, whether it's an isolated virus or directly from clinical samples. And again, the caveat is it really depends on how much virus is there. But especially these cases with where we can't find an etiological agent, those are the best ones for us to start with, because at least we have an idea what's happening in the field, and when we find something, we can try to make sense of it. We do a lot of metagenomic sequencing to profile herds as well as samples used for live exposure. So, feedback's very commonly used for PD, Delta Corona, PRRS, rotaviruses, and a lot of vets have questions, what else am I feeding back to animals? So we can do deep sequencing to make sure there's nothing that we don't want to be spreading around. In terms of cost, again, $300 a sample. So it's a little higher than Sanger sequencing, but if you consider the alternative, running multiple PCRs, histopath, VI, that bill can easily reach up to $300. So my opinion is we need to use this much more often to really get a better understanding of what's going on in our pigs and a more comprehensive understanding of the swine virome. So I'd like to thank collaborators at K-State, South Dakota State, Iowa State, as well as the veterinarians that have submitted these samples and made all this possible, as well as the Pork Board for funding the development of the methodology, as well as the Lettuce and BI for funding the sequencing project this past summer.